This video was prepared to be a review for you of how the adaptive, also known as specific immune system, functions. Now, the first thing that you need to recall is that there are three kinds of cells involved in this system. All three kinds of cells are specialized lymphocytes, so they're produced by the process of hematopoiesis in the red bone marrow. There are the cytotoxic T cells and the helper T cells, and then there are also the B cells. All of these cells must gain immunocompetence before they are able to do their functioning. The cytotoxic and the helper T cells gain their immunocompetence in the thymus gland, whereas the B cells gain their immunocompetence in the bone marrow. Immunocompetence involves a maturation process whereby they become able to recognize and tolerate self-antigens, antigens that identify your own body's cells, and all of them must be able to recognize a specific foreign antigen. There are all different kinds of T's, cytotoxic T, helper T, and B cells, and different groups of these cells are able to recognize and mount an immune response against different foreign antigens. So we can attack many different types of pathogenic material that may enter the body. In order for them to actually function as immune cells, though, after they've gained immunocompetence, they must experience an encounter with their target antigen. This encounter is a complex process of a specific recognition, and it is followed up by a counter recognition process that we call co-stimulation. Only after these events have happened do they actually trigger an immune response. This slide from the Nobel website gives us an overview of the entire process of active specific immunity. In the slides to come, we'll look at it as a step-by-step process to describe in a little more detail the various things that are happening throughout the process. The process begins when some antigen, maybe a virus or a bacterium, enters the body's tissues. A resident innate immune cell in the area, like a macrophage or a dendritic cell, finds it and engulfs it by phagocytosis. The pathogen is digested by the cell, and its particles are displayed in combination with the marker protein MHC class II on the macrophage's surface. A circulating T helper cell that happens to be sensitive to this particular antigen recognizes and locks on to this MHC antigen combination by use of its receptor. A reciprocal signal now from the macrophage to the T helper cell, called co-stimulation, serves to activate this T helper cell. Now the activated helper T cell undergoes a series of mitotic divisions that we call proliferation. Proliferation results in a population of clones that are all responsive to the same antigen. Some of the clones then specialize into effector T helper cells, and others become memory cells that will circulate in the body for weeks, months, or even years. These active effector T helper cells now go to work mobilizing the troops. Some of them look for B cells that have also engulfed the target antigen and are displaying antigenic particles on their surface with MHC class II. The T helper cell recognizes it, locks onto this display with its receptor. After co-stimulation takes place, now it activates the B cell. The B cell responds by proliferating. The mitotic divisions again are called cloning, and some of the clones are memory B cells for long-lasting immunity, but many of them become plasma cells. Plasma cells are antibody-producing factories. Antibody molecules are Y-shaped structures that have various different roles to play in the process of destroying and clearing the antigen from the body. Now is a good time to remind you that there's another name for activated T helper cells. They're also called CD4 cells. These CD4 cells, in addition to activating B cells, can also activate cytotoxic T cells. In this case, illustrated here, the T helper cell has activated cytotoxic T cells that are also sensitive to the same antigen. So the activated cytotoxic T cells are on the alert for any of the cells of the body that have been infected by that antigen. And so in this illustration, we see a self cell that has been infected, and we see that the cytotoxic T cell is using its receptor to recognize this altered self cell because of its display of antigenic particles in combination with MHC class 1. After co-stimulation, this will result in the process of proliferation, cloning, whereby the cytotoxic T cell produces many daughter cells, some of which become memory cells, as we've seen earlier, and others become active cytotoxic T lymphocytes. These active cytotoxic T lymphocytes have a number of different weapons that they can use to destroy infected body cells. One of their weapons is a molecule called perforin, which directly perforates the plasma membrane of the infected cell and results in lysis or rupture of the cell, as we can see in this picture. Now we can follow the process all the way from beginning to end. So an antigen of a particular type enters the body's tissue, probably many copies of this particular antigen. Some cells of the innate immune system, such as dendritic cells or macrophages, will encounter copies of the antigen. As phagocytes, they will engulf it and digest it within the cell and display particles of the antigen on their surface in combination with MHC class 2. Circulating T helper cells that are sensitive to this particular antigen will encounter it and will lock onto the display using their receptors. This locking on is followed by a co-stimulatory signal from the dendritic cell or macrophage that serves to activate the T helper cell. The activated T helper cell undergoes a process of proliferation, producing many generations of clones, some of which become memory T helper cells, but many become effector T helper cells, which we also call CD4 cells. These CD4 cells circulate throughout the body with the purpose of activating the other arms or the other portions of the adaptive immune system. Some will activate B cells. In this illustration, part B, we see an activated T helper cell that encounters a B cell that has also engulfed and displayed a particle of this same antigen. The T helper cell will lock onto the display, will co-stimulate the B cell, which will result in activation of that B cell. The activated B cell undergoes the process of proliferation, producing many daughter cells, some of which are memory cells, but many which will be plasma cells that turn into antibody-producing factories. The antibodies that they produce have many different ways of immobilizing and clearing the antigenic particles from the body's tissues, any extracellular spaces. Others of the activated CD4 cells will serve to activate CD8 cells, the cytotoxic T cells. These CD8 cells, as they're circulating, will be looking for any body cell type that has taken in this particular antigen and have been infected by it. These altered self cells are displaying particles of the antigen in combination with MHC class 1. Remember, this signal says, "kill me," and that's the response of the cytotoxic T cell. When the cytotoxic T cell encounters this altered self cell, it becomes fully activated, undergoes a process of proliferation, producing some memory T cells and many activated cytotoxic T lymphocytes. Cytotoxic T lymphocytes have several different means to do so, but their response is to kill the infected body cell. As we can see in this picture, the infected body cell has had holes punched in it, and the cell is rupturing or undergoing lysis. Meanwhile, the memory cytotoxic T cells, memory B cells, and memory T helper cells will continue circulating throughout the body for weeks, months, or even years to provide long-lasting immunity. Not shown in this illustration, but also present in the process, are a type of effector T helper cells that are called suppressor cells. Their job is to stop the immune process when it's done, when the antigen has been cleared. The whole process of providing innate immunity is very energetically expensive, and so the functioning of the suppressor T cells is also very important so that the body can return to normal.